So let's go over some of your vocab for today, and then we'll start doing some Punnett Squares together. So first of all, we have a lot of vocab, but I've got some tricks to help you remember them to make it go a little easier for you. So my first one is genotype and phenotype. So think about how the words start. So geno means genetic makeup. So if I ask you what an organism's genotype is, I'm asking you what alleles or letters do they have. So these are all examples of genotypes, the letter or allele combinations that you can get. Phenotype, on the other hand, pheno is physical. So if I ask you what an organism's phenotype is, I'm asking you what do they look like from the outside, not genetically. What is their physical appearance? This could be what color are they? What traits do they have? Do they have dimples or no dimples? Things like that. And then down here, these terms should sound familiar because these prefixes we've been using all year. So we have the terms homozygous and heterozygous. Remember the prefix homo means same and hetero means different. So homozygous means both of your alleles are the same. If I say homozygous dominant, that means you have two dominant alleles, two uppercase letters. If I talk about homozygous recessive, that means both alleles are recessive. They're both lowercase letters, but regardless, homo means same. So pay attention especially to the word after it because it lets you know whether they are both uppercase or both lowercase letters. And then heterozygous, remember hetero means different. So heterozygous means your alleles are different from each other. You have a dominant and a recessive. So when we do Punnett squares, essentially what a Punnett square is it's a way for you to calculate the probability of what traits or features, genotypes or phenotypes, the offspring might have. So we're going to do a quick example if you kind of see how it works. We're going to use Mendel's flowers as an example. Now remember, for every problem that you do, you need to choose a letter. You need to choose any letter you want. Like I said, just make sure it's different, uppercase and lowercase, so it's easy to tell which one's which. Choose one letter per problem. So if we go back to the Mendel's experiments from last week, remember he learned that in his pea plants, purple was dominant to white. So we know that anytime I talk about the purple allele, I'm going to use an uppercase letter, and the white allele, I'm going to use a lowercase letter. So what we're going to do is use a Punnett square to help us calculate the probability of what our offspring would look like if the female part of the flower was homozygous dominant. That means she has two purple alleles. Her phenotype would be purple. On the outside, she would look purple because she has two purple alleles. And let's say that the male part of the flower is heterozygous. He's got one of each. So he is also going to have a purple phenotype, physical appearance, because remember when you have a purple and a white, I know my dominant one covers up the other one. Now when we do a Punnett square, simply you're going to draw kind of like a window box that looks like this. We have mom and dad's alleles over here. You don't have to write them at the top like that, but here's how it works. Remember, we talked about this idea before that mom has two alleles and dad has two alleles too. Now, mom's pretty easy because we know what allele she's going to pass on to her offspring because she has two of the same kind. With dad, though, it's a 50-50 chance. His sperm cells could either have the dominant allele in them or the recessive allele. So what we want to do is we take a look at our square and we want to split up our letters. So mom has two dominant alleles. I need to take her alleles and then I'm going to put them on the left-hand side of the box. Okay. So I'm going to take her letters and I'm going to put them and I'm going to split them up to show that some of her egg cells will have that one, some of her egg cells will have that one. I know they're both the same, but don't worry about that for now. For dad, I'm going to put dad's alleles on the top. So you guys will notice that parent alleles always go outside the box. So dad has a dominant and dad has a recessive allele. He's got one of each. Now we fill in the boxes. What's inside the boxes represents the possible combinations of the offspring. Now you'll start in the box one and just kind of say this to yourself as you do it. You always pull an allele from the left and you pull an allele from the top. and you bring those two together. So in my first box, and you're always going to have 2 alleles in each box, because remember, individuals have to have 2. So this is one possible genotype that the offspring could have. I go to my next box over here. I always pull the allele from the left, and I always pull directly from the top, and I bring those 2 letters together. Now, as a rule of thumb, if you have both, you generally write the dominant one first. Over here in this box, I always pull an allele from my mom, and I pull an allele from my dad. You got to get one from each parent, right? That's why you pull from the top and pull from the left. This box over here is kind of a big reach. I got to pull from the left, and I need to pull from the top and bring these 2 letters together. So when you're all done and you look in your boxes, because I have 4 boxes, each of them is like a 25% chance. So if I want to know what my offspring might look like from here, I would say that half of them would have the homozygous dominant genotype, and half of them would be heterozygous, right? You can say 2 out of 4. I have 2 boxes out of 4 boxes. That's fine too. So this would be what my genotypes would look like. But if you go back to the box and you look at phenotypes, if I want to know physical appearance, here's what I would understand. These have 2 purple alleles. These will be purple. This will be purple. If I have one of each, that will be purple, and this one will be purple too. So if I want to talk about phenotypes, 100% will be purple. That's the phenotype. So you guys may be choosing to do your notebook entry. So let's jump to your notebook entry now, and then you might already know how to do it. If you do, go right ahead and do your notebook entry. If you're not so sure, follow along with me and I'll go through it with you. But you can do this, you can do it on paper in your notebook, or you can do it on directly on the document if you want and make your own copy. So I'm going to do this on paper just because it's a little bit easier for me. This is the same exact thing that you guys have. So my first problem says, I'm going to let Leah take over here. That's my daughter. All right. So for my first problem, what I'm going to do is I'm going to cross together Ocean and Lucy. Remember, they were both heterozygous for this trait, for the tail color trait. Now, one thing we want to do, they're using T's in this example, which is cool. The dominant allele is having a blue tail, and the recessive allele is having an orange tail. So think about what my letters mean. So here are Ocean and Lucy's genotypes. We're going to put those into the square, and then we're going to complete our square. So remember, here's how it works. So we've got Ocean and Lucy. Remember, they have 2 alleles, so we have to split the alleles up. It doesn't matter whether mom or dad are on the left or the right, but you have to take those 2 alleles and you have to divide them up. The other parent has the exact same 2 alleles. You put those across the top. And then you do that same thing we talked about. You always pull from the left and you pull from the top, and you pull those 2 together. So I'm going to pull from the left and